talk today about within host modelling. So I'm going to hand over to the first speaker. So we have Grant Lythe from the University of Leeds. Thanks.
Thank you. So I'm up in Leeds. I'm not down in Cambridge, obviously. It's a shame we can't meet in person. But on the other hand, I would have spent the morning taking the train, changing at Peterborough, going around via Ely, getting off at Cambridge, walking across to the Isaac Newton Institute. So there are some benefits to doing things remotely. So the F in the title is for Francisella. I'm going to talk about infection with Francisella tularensis. The other word is a place in California where the bug was first discovered. It's actually a bacterium, but it behaves in many ways like a virus. So the modelling is of current interest, you might say. So this project came about because Joe Gillard came to a meeting actually in Cambridge. The BSI has a mathematical modelling affinity group, and one of the first meetings he came to in Cambridge, that's Joe Gillard from DSTL. Tom Laws is a immunologist from DSTL that we've spoken to on many occasions, now a visiting researcher at the University of Leeds. The data that I'll show later on is Roman's data. And so the three people in Leeds that are part of this project are Martine, Carmen, and Jonty. Let's have the next slide. So I'll talk a little bit about computational model and analysis, which is a birth and death process with catastrophe. So it's only the last word that you might not be familiar with. There's some interesting mathematics in that, and also comparison, including ABC with Roman's data. And depending on how fast I go, perhaps in the half hour, I'll have time to talk about Ebola as well at the end of the talk. Let's have the next slide. So for people who know, it's a gram-negative bacterium, and remarkably, it has a very low infectious dose, and as well as being very dangerous, that's why the interest of people like DSTL was provoked. You can imagine it being sprayed in aerosol over a city and having devastating effects. So literally, it is believed that a handful, let's say 10 or a dozen of bacteria could be inhaled in one person and enter alveolar macrophages. So the bacteria can enter various cells. The ones that are in the alveoli of the lungs are called alveolar macrophages. And what happens, or what doesn't happen, is that the immune response that you would normally expect when something like that gets into your lungs doesn't seem to happen at all for at least 24 hours, perhaps 48 hours. Let's have the next slide. So here is what we believe does happen. So let's just go across the top row. There are 4 little diagrams. So each one of those represents one alveolar macrophage. And inside, on the left, on the top left, there is a bacterium inside a phagosome. So macrophages eat things. Their job is to eat bacteria and normally gobble them up, destroy them. So initially inside a phagosome, but what happens is that the Francisella tularensis bacterium is able to escape from the phagosome into the cytosol, into the main part of the interior of the cell. And in there, it makes copies of itself. It's a bacterium, it's actually a cell, it divides, and it can make many copies of itself, eventually causing the macrophage to burst, to rupture, we say, and a burst of bacteria are released back into the extracellular medium to infect probably other cells, or, well, I'll come back to what else might happen. But first, let's talk about the host cell a little bit, the macrophage. So there are various states that such a cell can be in. There's the standard, you might say, resting state, which in the middle part of the diagram, or the middle row of the diagram, is represented as green. Now, what happens, again, unusually, when a cell becomes infected by a Francisella tularensis bacterium, it actually goes into a suppressed state rather than an activated state. So instead of telling its neighbors to beware that something dangerous has entered the body, they actually, by virtue of something called TGF-beta, tell their neighbors also to be in this suppressed state, we call it. Okay. On the other hand, on the right-hand side in the middle, you can see what does sooner or later happen, which is that the macrophage can be moved into a activated state where it's producing interferon gamma and so on, which is actually doing what will finally get the immune response started. And that happens actually not because the bacterium goes inside a cell, but because associated with the rupture, top right-hand side, when the bursted bacteria are released, there are various chemical signals that cells recognize as something bad going on finally. Okay, so, and then in the middle, you can see the idea that when a macrophage ruptures, numerous, in fact, it can be hundreds of bacteria are released at a time. Some of those immediately infect more macrophages or more target cells in the same organ where they are, initially the lung. Some of them move to other organs. There we've written liver, spleen, kidney, etc., because those are the things where, as you'll see, measurements have been made. Or some of them actually won't survive for long outside the intracellular medium. If you just press sort of the next slide, this releases another sentence. It just repeats more or less what I say. Instead of producing inflammatory cytokines, the first infected macrophages produce anti-inflammatory TGF-beta. And one more click. And again, just to repeat, the released bacteria can migrate to another organ or again infect macrophages in the same lungs, supposing that the medium of infection was by an aerosol and they arrived in alveolar macrophages to start with. And next page. So you might be expecting me to start off by writing a set of ODEs like this, which I believe everybody in the audience knows and loves. And if you press again, you'll get the right-hand side. So you have free infectious agents that decrease at a rate proportional to the product of T and V, where T is the target cells. Okay, that RTV part is present in all three equations. All three types of things can die with some rate. If you press the next thing, okay. Most importantly, I've put it in red there because there's an underlying assumption when you're using these ODE models that the rate at which new free infectious agents are produced is some constant multiplied by the number of infected cells. Okay, and that's not appropriate here because, like I said, there's a very small number of initial bacteria that enter a relatively small number of cells in the first cohort. Nothing happens for quite some time, a day or two, and then suddenly lots of bacteria are released. So we need a different type of model. If you press it again, it's just showing the diagram, which is what I've said. So it's not the case. So there are three little pictures there at the bottom, and in the first case and the second case, no bacteria, no new infectious agents are being released, and then at the end, a whole lot are released at once. So instead of that standard ODE type of model, we're going to do something different. You can press on to the next page. Just to repeat what I've said, that it's an extremely virulent pathogen, okay? And what we're going to do, instead of thinking of the number of new infectious agents being proportional to the number of infected cells, the rate at which new ones are produced, we're going to think about what goes on in one cell at a time. So there's a different type of simplification, which is that in those cases where the bacteria are inside cells doing their stuff and not releasing anything, then each cell is a story unto itself. The population of bacteria inside each cell is independent of the other ones. Okay, next slide. Okay, so what we're going to end up doing is something called a birth and death process with a catastrophe. So inside one cell, the number of bacteria is something which can increase by 1 at a time. When a bacterium divides, you get an extra one. They can also die sometimes, so you get one fewer. That's the standard birth and death process, and that carries on until some time, which from the point of view of counting the number of bacteria inside a cell is a catastrophe, because the cell ruptures, dies, that's the end of the story for that cell, and suddenly the number collapses down to 0. So it's an interesting type, or an interesting extension of the standard mathematical idea of a birth and death process. Let's have the next slide. Okay, so I'll come back to the birth and death process. Let's talk about the agent-based computational model, where we can try to put the kitchen sink in if we want to, meaning that we can put in everything that we believe is a correct description of what's going on. We can literally have a list of macrophages and a list of bacteria. Each one has an identity, so we know where it is. We know what its attributes are. So the attributes of a macrophage are, as I've mentioned, the state of activation, the cohort counter, I might have time to come back to that, the number of bacteria in its phagosome at the time, well, in phagosomes, 1 per phagosome, not just the number of cytosolic bacteria, so the number of bacteria in the main part, the cytosol of the cell, but actually which bacteria they are. So you can have a list associated with each macrophage. Okay, and also where in the body the macrophage is. So we only have space in that sense, which organ the cell is in. So yes, repeating what I said about the types of macrophages, or the states that they can be in: resting, suppressed, or activated. Okay, if you click again, then you get the information about the macrophages. So they begin all in the resting state. When a bacterium goes into a macrophage, it automatically goes into the suppressed state. How can they get into the activated state? They detect host damage caused by rupturing macrophages, things called DAMPs and PAMPs, if you're familiar with that terminology. Okay, and once the macrophages are activated, they do kill the bacteria that they phagocytose, and they also secrete interferon gamma that provokes the activation of neighboring macrophages. So already the hypothesis, or the story that's believed to explain the lack of an immune response for the first at least 24 hours in this case, is that the suppression is winning over the activation, at least for that time. Next page, please. Okay, so let's go through a little bit more the actual computational model. First of all, any bacterium in a cytosol of a host macrophage has a birth rate, beta. So I say reproduce means division in this case, and also death rate, mu. The rupture is defined by what you might think of as, thank you, a hazard rate. Okay, so each macrophage has a certain number of bacteria in its cytosol at the particular time. At that time, you can calculate a rate, which is just delta times the number of bacteria, and that's the probability per unit time of this catastrophe happening. Catastrophe from the point of view of the cell, where the cell dies, ruptures, releases all its bacteria, and those bacteria can go on and do something else. So we're not saying that there's a certain time that this must take. We're describing this as a stochastic process with its own rate. Next bullet point. So the bacteria released, as I've said, either reinfect macrophages in the same organ, there's a rate rho associated with that, or migrate to a different organ. Okay, one more bullet point, thank you. And the suppression or activation are described in the computational model by two dimensionless functions of time. Let's go on to the next slide. Sorry, one more bullet point. So initially there are N bacteria, capital N, and M macrophages. So N is usually a small number, fewer than 100. M, capital M, is a large number, 10,000 or more. Okay, now We've seen this diagram before. The first row across is what we're modeling when we're thinking about one macrophage at a time. But when you put the whole computational model together, you can see how this whole story develops, spreads around the body, and unfortunately for mice, always, as far as I know, and for people quite often, the result is the death of the mammal. Okay, so to actually do the time stepping, press the there you go, thank you. To actually update everything. So you've got a list of macrophages, you've got a list of bacteria, they've all got their attributes. You have to decide in the stochastic simulation algorithm, the Gillespie algorithm, what is the next event from a list of possible events. So that list, in principle, is very large. First of all, there are 8 types of events: phagocytosis, escape, replication, rupture, death, migration, suppression, activation. Things that can happen that affect the state variables. Secondly, I've just shown 3 spatial locations here, the lung, liver, the spleen. So the first paper that we wrote, 2014, the data was there were only measurements in those 3 places. Okay, so that's already 24 types of event. And you should think, when you're doing the Gillespie algorithm, of the unit interval being divided up into subintervals of different lengths. The length corresponds to the rates associated with that event. And you should think of throwing a dart or something that's equally likely to arrive anywhere between 0 and 1, and where it happens to fall, that's the next event. There comes a time when you're doing this type of simulation and scaling it up to something physiological, where you have millions of possible events, and you probably abandon the Gillespie algorithm and do tau leaping instead. But nevertheless, you still need to keep in mind that there are 8 types of events multiplied by a number of different organs. And even after you've decided which of those colors on the bars is the next event, you have to decide which particular macrophage and which particular bacterium that event occurs to. Okay, let's go on to the next slides. So back to the mathematical idea, which is the simplification, just thinking about what's going on inside one cell, one infected cell at a time. So Markov process, where the integer in the middle of the circles is the number of cells, number of bacteria inside one macrophage. So you would start off at number 1, when one bacterium has entered the cell, initially inside a phagosome, escaped from the phagosome, is now in the cytosol. Okay, it makes copies of itself. Sometimes those new bacteria die, so you can hop to the right or to the left for many hops possibly, until eventually the whole story comes to an end when there's a catastrophe and the process goes down to H. Okay, well, usually that's how the story ends. It's also possible for the story to end because the process makes it down to state 0. Next slide. Okay, so in terms of the Markov process description, there are only 3 events that can happen. So if you're in state n, you can go to n+1, you can go to n-1, or you can go to h. And in each case, the rate is proportional to x. So it's a fairly simple Markov process, but it's not technically a branching process, because it's not the case that each bacterium is independent, because the catastrophe affects everybody at once. Nevertheless, you can, if you go on to the next slide, you can define a survival probability. So that's the probability that the catastrophe hasn't happened yet. And because the probability that the catastrophe happens in the time interval, the next time interval of duration delta t, is proportional to delta times the current value of x, you can write down a relationship between the differential of s, ds dt, and the mean of x, which is written there. And in fact, you can go a bit further and you can write down a self-contained closed differential equation for s. Okay, And the right-hand side of the differential equation at the bottom left is a quadratic, which is drawn there, and it has two intersections of 0. One is A and one is B. A is less than 1, B is greater than 1. So remember the parameters were mu, which was the death rate inside a cell, beta, which was the division rate inside a cell, and delta, which is the catastrophe rate. So if you're good at this, you can already notice that the quantity small a is the probability that the cell wins the battle, that the bacteria are eliminated from the cell before the cell ruptures. Let's go on to the next slide because I'm already running out of time. Okay, so you can actually go ahead and write down the generating function like you would for a branching process. It's not a branching process, but nevertheless, you can solve for that probability generating function, and you can extract all sorts of useful information from that. Let's go on to the next slide, since I don't have time to talk about the maths. So if you look at there are 3 graphs now. Top left is the survival function. So it starts off at 1. So the probability that macrophage is alive at time 0 is 1. Okay, as time goes on, it decreases. So there's a blue line and a dashed line. The blue line is when there is intracellular death, and the dashed line is when there isn't. So if there's no intracellular death, then it's just a birth process which carries on until the catastrophe happens. Otherwise, there's a small probability that the birth and death process ends up at 0 rather than the state we called H. Okay, the middle one, on the one hand, is easy. It's just a derivative, or minus the derivative of the first one. So that's the probability density of rupture times. Okay, it's also, because they are related, it's the mean number of bacteria as a function of time, which starts off at 1, increases, and then decreases. Why is it decreasing? Because some of the cells are dead. Okay, and then you can also say, what is of, now you should think about an ensemble of cells, what is the rate of release of bacteria? Okay, so that's getting back to why we're not using the differential equation description. So that goes up and then flattens off, interestingly. All right, let's carry on to the next slide. Okay, so the first thing we can do is compare this idea, which is, remember, just mathematical birth-death process with catastrophe, to the agent-based model, which is a fairly complicated computational model. So suppose I have initially 30 infected macrophages. Okay, so now my survival function is a number which starts off at 30. The number of macrophages at time t starts off at 30 and goes down in steps until it gets to 0. So on the left and the right, there are 2 realizations, exactly the same code, exactly the same parameters, just a different realization. It's stochastic, okay? And the solid lines are from the theory. So the survival function is easy to understand. The lower 2 graphs with the blue dots, they're telling you how many bacteria are released each time a cell ruptures. Okay. So that works out quite nicely. Let's carry on to the next slide. I'll forget about that because I'm running out of time. Let's carry on to the next one. Okay, let's do the data because that's nice and interesting. So these types of experiments, by the way, are extremely difficult to do because you can only manipulate Francisella tularensis in extremely controlled conditions. Okay, but you can infect mice and see what happens to them in the few days before they succumb. And you can measure, in particular, count literally the number of bacteria in various organs. So the infection is through the lung. So there are measurements in the lung, which are in blue, you can see there, and other organs in different colors. So let's go on to the next slide so I can explain a little bit better. Okay, so we have these parameters in the model, and we can do Bayesian computation to try to learn the values of these parameters. So I won't have time to go through all of them, but the most interesting one is the beta. So that's the division rate, the rate at which a bacterium inside the cell of a macrophage divides, or the probability per unit time for dividing in the next time interval. So you might have noticed in the previous examples when I gave, I chose a value for beta, which was 0.15. And in the first paper we published on this, we used that value because in the literature it said that the mean time for division of a bacterium inside a cell had been estimated to be 6 hours. So 1/6 hours is about 0.15. Okay. Now, astonishingly, when Jonty did the ABC for this model, the posterior distribution, which you can see there on the bottom left of beta, was beautifully peaked just about 0.15. It looked like we cheated, but it's honest to God, that's what it was. If you remember on the previous slide, the data wasn't very impressive. You wouldn't think you can get much out of data like that. But if there's one thing you can get, it's an overall growth rate, and that's beta. Just quickly on the right-hand side, you can see some other parameters. So M, remember, was the number of macrophages. Rho was the rate for migration to a different organ. And gamma, rho is the reinfection rate in the same organ. Gamma is the rate of migration to a different organ. Okay, so in that case, we can't fix those parameters from the data, but there's clearly a relationship between them. So what we are actually fixing, when you think about it, is the probability of migrating before reinfection of a different cell in the same organ. Okay. All right, so I'll just do a quick summary. Mouse infection model, agent-based computation, mathematical analysis. And the story is that the bacteria enter the host cells and proliferate inside, eventually destroying the cells. Okay, so the analysis is based on a stochastic model, which extends the birth and death process by the occurrence of catastrophes. And the ABC was very successful in this case. All right, so I'm guessing I don't have time to do Ebola.
I think we have a couple more minutes if you just want
Couple more minutes? Okay, fine. Let's just do Ebola. So, well, it probably needs no introduction to this audience. So it actually is the virus this time. Okay. And the release of the new infectious agents, the virus or viral particles, occurs by budding. So it does, in principle, make sense to describe the rate of production of new virus as being a constant multiplied by the current number of infected cells. But even then, the plot thickens. Let's go on to the next slide. So what is believed to happen, and what we tried to build into the model in this case, is the idea of an eclipse phase when, a little bit like what we saw in Francisella tularensis, the virus is inside the cell, but the cell hasn't started to release new virus yet. Okay, and within that eclipse phase is a co-infection window where you can get reinfection of the same cell. And if you see a series of green circles with arrows and orange or red circles with arrows, then that's a mathematical trick we're using where instead of having an exponential distribution of time, we construct a more interesting, more realistic distribution by a sum of exponentials, by inventing these, you might say, imaginary intermediate stages. Okay, so now we've got, as well as rates, we've got a number of stages, which are parameters that we'll try to fit from ABC. Let's go on to the next slide. Okay, so just as a quick summary, there's a co-infection window, there's an eclipse phase, there's an infectious phase. Okay, this was in collaboration with, as well as DSTL, Public Health England, and Julian Hiscock's group in Liverpool. Still going, by the way. Okay, so let's go on to the next page. Thank you. Okay, so what we got out of this so far is that the eclipse phase is about 30 hours. Okay. After that, it releases virions about 13 per hour for a time of about 83 hours. Okay. So what that means is that, again, the number of virions produced from one initial infection in one cell can be enormous, about 1,000 this time. And if you care about reproductive numbers, it could be about 600. Okay. So there are various interesting assays which I don't have time to talk about. I'll just mention that part of the interest here is in something called defective interfering particles. So viruses have the weakness that they mutate a lot, and sometimes they produce defective versions of themselves, and those versions of themselves can compete with the original virus, okay? So that's bad for the virus, good for us if we're trying to kill the virus. So if we can find a way to sort of favor the defective particles, or perhaps deliberately introduce them, that might be some sort of antiviral method. Okay, let's carry on to the last slide. So on the right, you have Jonty Carruthers, who's the PhD student who did most of the work, especially the ABC stuff that I've talked about. That's him on the left-hand side of the photo, the day of his viva. Martin and Carmen are in the middle at the back, standing against the door. So the people I need to thank, I already mentioned Joe and Tom and Roman from DSTL, the level 4 virology team as well. So recently on Nebula and other things, Laura and Alan have been talking to us. There's Isabel and Julian from Liverpool, Catherine Beauchemin from Ryerson in Canada. Also John Paul, who's a statistician at Leeds, or he was until recently, John Barr, virologist at Leeds. And, oh yes, I mentioned the BSI affinity groups. This still exists, the Mathematical Modelling Affinity Group. We normally would have had our meeting this month. Unfortunately, as you know, everything is unusual at best online. Okay, I'll stop there and hopefully take a question or two. Thanks for your patience.
Well, thanks very much, Grant. And give a round of applause, a virtual round of applause. So I think, unless anybody has any burning questions, perhaps we'll leave questions for all of the speakers until the discussion section at the end, if everyone's okay with that.
Good idea.
Yeah, okay, fantastic. In that case, we will move on to our next speaker. So hopefully, Raluca, you can share your slides. Fantastic. Okay, so I'm really pleased to introduce Raluca Eftimi from the University of Dundee now to tell us about her work. Over to you, Raluca.
Hi, thank you very much. Yes, right now I can share my slides. Okay. Okay, so basically, I'm gonna talk about a few projects that I've been doing over the past few years related to viruses, oncolytic viruses, and cancer. And I will only mention on the last slide my current work on COVID-19 and viruses, and again, so the immune cells. So to start, well, I'm interested in oncolytic viral therapy. What are these?
Sorry, Raluca, can I interrupt you? I'm not sure we can see your slides. Are you seeing, I'm just seeing a blank screen at the moment.
Oh.
Do you know, I think if
Can you see it now?
Yeah, I think you might need to leave in Zoom if you're using Acroread. I think you have to leave it like that. Yeah.
Okay.
Great. Sorry, carry on.
Sorry. So oncolytic viruses are basically viruses which are genetically modified to infect selectively and replicate inside and kill tumor cells, while they're not harming healthy cells. For a very long time, it was considered that the therapeutic efficacy of these viruses depends on the direct viral tumor interactions. And researchers thought that the immune system impairs virus delivery and virus spread. So there was this virocentric point of view related to these oncolytic viruses. So immune system is bad, it should be shut down. However, more recently, oncolytic viruses, they've noticed that they can actually induce anti-tumor immunity, which of course is a key factor in treatment outcome. So there is a shift between virocentric and immunocentric point of view. So now immune response is not bad, but actually could be good too, and it can help anti-tumor therapies. basically, you have the oncolytic virus, gets into the tumor tissue, destroys some tumor cells. At least so you have an immunogenic cell death that leads to the release of tumor-associated antigens. You might also have some pathogen associated molecular patterns, so PAMPs, DAMs, damage-associated molecular patterns. They are recognized by the dendritic cells, they trigger the adaptive immunity, so T cells, and then they kill the tumor. So that's the ideal picture. Effective cancer immunosurveillance does require, so why do we care about this oncolytic virotherapy and immunotherapy? Cancer immunosurveillance requires the expression of tumor-associated antigens that can induce the expansion of effector cells. The good thing about this oncolytic viruses is that, you know, the viral oncolysis then can lead to the release of the whole set of these tumor-associated antigens. One such virus that has been shown to do this is the vesicular stomatitis virus, which I'm going to talk about after this. The production of the cytokines and danger signals by the infected tumor cells infected with the oncolytic virus should reinitiate the anti-tumor immunity. So oncolytic viruses actually can help the anti-tumor immunity to obtain a much better tumor outcome. But there are many other aspects of oncolytic viruses. They can actually help induce tumor vasculature, shut down tumor necrosis, and so on. However, there is a bit of problem that we have with these oncolytic viruses. So they do trigger anti-tumor immune responses, but the immune response against the viral antigen, so against the virus, can actually overwhelm the immune response against the tumor-associated antigens. So we're still not fully understanding the delicate balance between immune response against the tumor versus immune response against the oncolytic virus. One cell that is very important in the antiviral immunity is the macrophages. Macrophages are also important in tumor dynamics. They play a central role in innate and adaptive immune responses. And they're also actually one of the most common cell types in solid tumors. For example, in breast cancers, they can form up to 50% of the total tumor mass. In general, increased macrophage infiltration is associated with poor patient prognosis. We also know that activated macrophages can kill cancer cells by themselves, directly or in an indirect manner through the recruitment of other immune cells. So the question is, so if macrophage infiltration is associated with poor patient prognosis, and if you have, if the activated macrophages actually can kill cancer, where is the problem? Why lots of macrophages inside the tumor means poor patient prognosis? The answer is actually in the fact that the macrophages are a very heterogeneous population of cells. The two extreme phenotypes are the M1 and M2 cells. M1 are the classically activated macrophages. M2 are the alternatively activated macrophages. M1 cells are involved in immune activation. They have anti-tumor property and pro-inflammatory properties, leading to tissue injury. On the other hand, the M2 cells at the other end of a continuum, they are involved in immune suppression. They have pro-tumor properties and they are anti-inflammatory. During tumor progression, there is a transition from a M1 phenotype macrophages, so classically activated, towards an M2 phenotype, so from an anti-tumor role to a pro-tumor role. Treatments can repolarize macrophages from an M2, from the pro-tumor phenotype to the anti-tumor phenotype. Okay, how are the macrophages and viruses interact? Well, so most of the tumor-associated macrophages are M2, but we've also observed M1-like cells in early tumors. Well, that's actually the general accepted idea. M1-like macrophages seem to enhance the virus-mediated activation of the anti-tumor immune responses. And they also seem to promote the antiviral immune responses, so they lead to virus elimination. M2 macrophages are associated with tumor angiogenesis, metastasis, suppression of the anti-tumor immune response. So they might actually, they suppress the antiviral immune response, therefore they promote viral proliferation and oncolysis. However, the situation is slightly more complex because, so the role of macrophages in oncolytic virus therapy, it's understudied and also potentially controversial. For example, so there's this paper published in 2016, tumor-associated macrophages and oncolytic viral therapy, friend of or foe, and the authors looked at a number of tumor types, the macrophages polarization, and the oncolytic virus in some cases, so the oncolytic virus used for those treatments. I'm not going to focus on all these different types of viruses, but just to mention some studies. For example, Tan and his collaborators noticed that the tumor-associated macrophages, irrespective of their initial polarization rates, they can enhance the effect of measles, the oncolytic as an oncolytic virus, and mumps viruses by acquiring an anti-tumor phenotype. So the virus helps the repolarization of macrophages from a pro-tumor towards an anti-tumor. Other studies have actually showed that tumor-associated macrophages impede this oncolytic viral replication of herpes simplex virus, adenovirus, and vesicular stomatitis virus. So there is lots of unknown when it comes to the interactions between oncolytic viruses and macrophages. And the goal is to use a mathematical model. Actually, there'll be a few models to identify the mechanisms which could lead to this polarization or repolarization of macrophages from M1 to M2 and from M2 to M2 to M1 in the context of cancer development. The focus will be on a vesicular stomatitis virus. This is I forgot to show you a picture. It's basically a negative stranded RNA virus that infects a variety of mammalian insect cells. So it's endemic in horses, for example. It's endemic in mammals in Central America and North and South America. In humans, it can cause mild disease. It's being liked as an oncolytic virus because of a lack or a very low pre-existent human immunity to this virus, to vesicular stomatitis virus. Cellular responses to this virus induce a type 1 interferon production. Obviously, viral replication occurs when cellular responses are suppressed. And the good thing with cancer is that in many cancer cells, antiviral responses induced by this type 1 interferon production, they are impaired. So virus can replicate very nicely inside many types of cancers. It has been used on B16 F10 melanoma. So it's a melanoma cell line in mice. So murine melanoma cells. I'm gonna show you a mathematical model or a class of mathematical models. There'll be all of ODE type because the data that we had access to is just temporal data. So we decided to start with the simplest possible models. So for the data, we had some data on tumor cell numbers. Again, so this murine melanoma line, and M1 and M2 macrophages, and the percentage of M1 macrophages and M2 macrophages. So they had been published by Chen and his collaborators, 2011. So we have the percentage of macrophages on day 7 and on day 14. Regarding tumor and vesicular stomatitis virus, we use a different data set. Unfortunately, there was no there was no study that looked at all these different interactions in one experimental setting. So we looked at data, again, the same tumor and vesicular stomatitis virus, so tumor growth data. For the mathematical model, so very simple interactions. We have the M1 cells, which can kill the viruses. M1 cells can kill the tumor as well. Viruses infect the tumor cells. M2 cells help tumor growth, and also they help viral proliferation. And there is this repolarization or polarization from M1 to M2, and then repolarization from M2 to M1 cells, depending on the tumor microenvironment. So model, very simple one. We're looking at uninfected tumor cells. They proliferate. They can be infected with a virus, and we use the saturated form to describe the fact that actually the virus does not interact with all tumor cells inside the solid tumor because of physical barriers, extracellular matrix, very tight space between, tight junctions between cells. So we prefer to use this saturated term to describe the fact that you have lots of viruses, but these viruses interact only because of the physical constraints inside the tumor. So we're not using a special spatial model. We're looking at a temporal model, so only OD, but this particular term here describes the restrictions given by the spatial structure of the tumor, restrictions in terms of viral spread inside the tumor. The uninfected tumors can be killed by the M1 cells and and this killing is reduced by the presence of M2 cells. And there is also the pro-tumor effect of M2 cells, M2 macrophages. So we have the infected tumor cells. The infection rate is dV. Delta I is lysis rate, and then again, infected tumor cells can be eliminated by the macrophages. The virus is introduced into the system at some time. So we have the killing rate, so the lysis, which leads to the release of a number of particles. So B is the burst size. The virus can also be eliminated from the system and can be detected by the M1 macrophages and killed. For the macrophages, we have these activation rates in the presence of the virus or by the tumor. There is the proliferation rates because macrophages can proliferate inside the tissue, or they can migrate towards the tumor tissue. Then these large terms here, they describe the polarization. So in the presence of tumor, there is a very low baseline polarization rate from M1 to M2 due to whatever cytokines in the environment, tumor microenvironment that there are. But also there is a much higher polarization rate induced by the tumor. And then there is the repolarization of macrophages from M2 to M2 to M1 due to this viral treatment. So viruses, actually the presence of the virus in some studies, experimental studies, it has been noticed that the virus can induce a repolarization from M2 to M1. So we're looking at data, we're comparing the model with the data and try to estimate the parameters. There is a pretty good fit to both data sets, which allowed us to estimate different parameters. It's not ideal, but that was the best we could do. We had some sort of baseline behavior of the model where the tumor is growing, reaches carrying capacity, and then this is associated by an increase in the percentage of M2 cells, which is what everyone expects. As tumor is very small, you have a very large percentage of M1 cells. As tumor is increasing, you have a high percentage of M2 cells. But here it is. So we're trying to understand the interactions between macrophages and viruses. And for this, we're going to look at possible, so some TAM-targeted therapeutic strategies, as identified in this paper in Frontiers in Biosciences in 2015, ways of repolarizing macrophages to ensure patient survival, where we can repolarize M2 macrophages towards an M1 phenotype, or we can block the recruitment of macrophages and monocytes to tumors. We can increase the antibody-mediated elimination of tumor cells by the macrophages, or we can suppress the survivals of these macrophages. I'm not going to focus on all these 4 different strategies. Next, so this was discussed only in the context of macrophages and tumor. Next, we're going to look at what happens when we have macrophages, viruses, and tumors. So first, we're increasing, the repolarization of M1 cells towards M2. Again, in tumors, you don't want to have too many macrophages, but M2 macrophages, because these are pro-tumor, so they help tumor growth. However, because you have the oncolytic virus, M2 macrophages are thought to actually help tumor because they suppress the antiviral M1 response. So we first looked at what happens when we are actually increasing, or when the tumor induces this fast M1 towards M2 polarization of macrophages. Well, the outcome is not great. So the tumor reaches carrying capacity. Eventually, there is lots of virus there, and the tumor is eliminated. But from an experimental point of view, this is pointless, because by then the patient is dead, the mouse is dead. So we are looking at now, this is panel A, and the elimination of the tumor, of course, is associated with a very high percentage of M1 macrophages. But again, it doesn't help in the context of experiments. Mathematically, okay, you get what you actually you expected. Panels B and C, we looked at what happens when we increase the repolarization induced by the virus. So from M2 pro-tumor macrophages to M1 anti-tumor macrophages. If we increase the repolarization and we keep the killing of the macrophages, of tumor cells by the macrophages by M1 cells the same, nothing happens. So the tumor still grows towards the carrying capacity. So you need a combination of multiple parameters. So it's not only the repolarization from M2 to M1, but this has to be associated by an increased phagocytosis, or actually so killing antibody-mediated killing of tumor cells by the macrophages. Otherwise, one approach at a time, so one treatment or anti-TAM treatment, it just doesn't seem to be working. Then we looked at what happens if we reduce the immune response. The reduced rate at which macrophages in one cells can eliminate uninfected tumor cells as well as the infected tumor cells. Okay, everything is beautiful. The tumor is, so the tumor decreases below its detection thresholds. And this decrease in the tumor level, although the tumor is not eliminated, but it is below threshold of detection. So the tumor might be there, however, we cannot actually see it, at least not in humans, where the detection threshold seems to be between 10 to the 7 and 10 to the 9 cells, according to a study published a few years ago. And of course, so the tumor is kept under control, and this is because of the M2 macrophages. The percentage of M2 macrophages, even if there is overall, there is a much lower number of macrophages inside the tumor. Why do I plot both number of macrophages and percentage? Because many studies, they look at number of macrophages or the percentage of macrophages as markers for prognosis, cancer prognosis. So I'm looking at both at what is the percentage of M1 versus M2 macrophages inside the tumor, as well as the total macrophages infiltrating the tumor. we also look what happens. Okay, when you reduce the immune response, the rate at which the macrophages kill the tumor, for a much shorter period of time, there is a reduction in tumor growth, in tumor size, but then it goes back to the killing capacity. Okay, we did a local sensitivity analysis to identify what are the parameters that are most likely to impact the tumor outcome. Obviously, so we looked at both in changes in the tumor size as well as changes in the virus levels. In both cases, it seems that you have the viral infection rate, so VV is very important. You have, of course, the tumor killing rate. There is also the burst size, so the number of virus particles released by infected tumor cells. That was for the tumor. For the virus, there is something very interesting going on here. So it seems to be a non-monotone relationship between the virus outcomes, the changes in the virus load, and some model parameters. And we did not actually continue with a global sensitivity analysis. We could have done it, but I don't know, for some reason we didn't do it. Okay. But the thing is, macrophages are not alone inside the tumor. So you also have CD8 T cells. Or you have so many other cells, NK cells, eosinophils, neutrophils, and so on. And you also can have CD4 T cells. So the next step was to look at, okay, what are the interactions between macrophages, part of the innate immunity, mostly associated, of course, they are also important in adaptive immunity, and CD8 T cells, part of the adaptive immunity. We ignored, in a sense, the relation between M1 cells and Th1 cells and M2 cells, Th2 cells, so between macrophages and CD4 T cells. And we only assume that, okay, CD8 T cells, they can also kill the viruses. And they are activated by the PAMs and DAMs that are released when the tumor cells are lysed by the viruses. So pretty much the same model as before, but we added an equation for CD8 T cells and for the viruses, and of course this impacts the viruses equation. We looked at what happens. So there was another aspect that actually we investigated here. When the virus is injected into the tumor, this might lead to actually changes in the killing rates, because before that, so the immune cells, both macrophages and CD8 T cells, they were not detecting this PAMs and DAMs, so the pathogen-associated molecular patterns or damage-associated molecular pattern released by the infected tumor, lysed tumor. So we assumed that we wanted to investigate what happens when we look at changes in this killing rates, and we obtained various tumor evolution patterns. Tumor is eliminated or the tumor is reduced for some time, just under the detection level or just above detection level. And this, we paid particular attention at the percentage of M1 cells and M2 cells associated with tumor control or complete tumor elimination, and as well as number of CD8 T cells. Again, because many studies suggest that CD8 T cells or the number of CD8 T cells that infiltrate tumors can be seen as a prognostic, can help us make a prediction about the evolution of the tumor. The thing is, for example, if you look here, yes, so around day 30, so tumor elimination is associated with an increase in CD8 T cells. But here, tumor elimination seems to be associated with a slight decrease in CD8 T cells. So the results are not always so we cannot actually make conclude that in all cases. It depends on the parameters and depends on the environment, macroenvironment. So we cannot conclude that tumor CD8 T cell level are always correlated with tumor evolution. So with decrease in the tumor, so tumor control or not. So just to sum up this bit, polarization of this inflammatory M1 or immunosuppressive M2 macrophages can enhance the oncolytic virus therapy, either through the antitumor immune activation or through enhanced oncolysis. M2 cells can have an antitumor role in the context of oncolytic therapies by inducing oncolytic replication. So even if you have enough M2 cells there, which you know that are pro-tumor and they're bad for the tumor, in the context of oncolytic viruses, they allow the replication of the oncolytic viruses, and therefore this might lead to a reduction in the tumor size. Then you also have the antitumor role of M1 tumor-associated macrophages. So it's a direct antitumor role, which can also be modulated by the oncolytic viruses, because these oncolytic viruses can lead to a repolarization of M2 cells towards an M1 cells, thus enhancing the antitumor role of M1 macrophages. It seems that we need both antitumor and viral antitumor immune responses for tumor control. And there is a very delicate balance between this M1 and M2 cells, especially when it comes to viruses used for treating tumors. And regarding the CD80 cells, so according to this mathematical modeling, and of course, these results are heavily dependent on the parameters, not always, this very high level of CD80 cells not always associated with permanent tumor elimination. Okay, the last bit, well, we have macrophages, we have viruses, and we have tumor cells. However, it is known that macrophages can be infected by these viruses. So, for example, there is a paper from 1989 which showed that vesicular stomatitis virus can actually infect macrophages. But at that time, it was not known what type of macrophages. So, which what is the phenotype of those macrophages that were infected with the VSV? This question has been answered in a very recent study, which was based on some master thesis from Appalachian University in the United States. The paper has just been published this month. It seems that this oncolytic vesicular stomatitis virus targets selectively M2 macrophages. So they don't really replicate inside M1 cells, they can replicate inside M2 cells. So we looked at, okay, a slightly modified version of the previous model. We said, well, let's ignore the tumor for now. Let's focus exclusively on the interactions between macrophages and oncolytic viruses. So we have a virus. It can be killed by M1 macrophages. It can infect M2 macrophages. So we have uninfected M2 cells and infected M2 cells. And then infected M2 cells release more viruses. And we wanted to again, use a simple mathematical model to try to get some sort of, basic understanding of the interactions between macrophages, M2 macrophages and M1 macrophages, as well as the oncolytic viruses. And this was a project that was published in April this year. It was a project with one of my PhD students, Nada Amwalem. We looked at asymptotic dynamics of the system. So we looked at conditions for the persistence of the virus in the long term, or for the elimination of the virus. And these three graphs describes possible evolution of the tumor, evolution of the system towards steady states where you have viruses and so coexistence of the viruses with the macrophages. Existence of macrophages and no viruses, or no viruses and no macrophages. So we looked at the conditions for the asymptotic stability and for the existence of, for example, here, we graphed the steady states for the virus, and versus PM is the proliferation of the macrophages, and there are different steady states that exist or are stable in some regions when the proliferation is less than the death rate, or the 1 over the half-life of macrophages, or proliferation is greater than that. And then Nada did a bit of local sensitivity analysis to get an idea of what's going on. So what are the effect of changes in the model parameters on the overall viral density? So, she looked at C is the burst rate. This is the repolarization rate of macrophages induced by the repolarization induced by the virus. Beta 2 is the virus infection rate. But we also did global sensitivity analysis, and we calculated the partial rank correlation coefficient. So it was a Latin hypercube sampling combined with PRCC. And we identified two parameters to have the greatest impact. So C is the burst size, this bit here. And so the first subpanel, panel A, and then the infection rate, subpanel D. Obviously, this is as expected. For the persistence of the virus, you need a large burst size and you need a high infection rate. And I'm going to stop here, mentioning the work in progress together with one of my MSc students, Mark. We're looking at investigating the role of macrophages and T cells on the persistence and the spread of SARS-CoV-2 and on the tissue destruction and repair, because you have M2 cells which are repairing the tissue, M1 cells secreting the cytokines that destroy lung tissue. But this is a story that will be told in the future. Thank you.
Great. Thanks so much, Raluca. So again, round of applause. Thank you very much. Another really interesting talk. Again, I think we'll hold the questions till the end, if that's okay. And so with that, we will move on to our final speaker. So my pleasure to introduce Farzad Fatihi China from the University of York. Thank you.
Thank you. I want to thank the organizer for giving me this opportunity to present my latest work on the modeling of SARS-CoV-2 infection and reveal the benefits and the risks of starting treatment earlier or later, what happens. So in this project, we are modeling intra and intercellular dynamics of SARS-CoV-2 infection. The virus is a positive trans RNA virus, although there are intra and intercellular models for other RNA viruses such as hepatitis C, but we really couldn't use those models for SARS-CoV-2 because each virus has its own aspects of life cycle and each virus acts differently. Thus, we developed one of the first detailed mathematical model for the intra and intercellular dynamics of COVID-19 infection. These models led us to study the dynamics of the virus inside cells and in patients' body with an unprecedented level of details. Also, as you know probably now, there's a lot of news about remdesivir, which was previously developed as a treatment for Ebola. It has been proved that it also works for COVID-19, but developing effective treatment protocols due to the rapid spread of these disease, due to the nature of this outbreak is really difficult. It's really difficult to do, but the theoretical model that we have developed in this work, they will let us to study the dynamics of the virus in patients and also to explore the effects of treatment on patients with different levels of immune responses, such as healthy or immunocompromised patients. As I explained briefly in the previous slide, so the aim of this work is to first develop an intracellular models which contains all the necessary process of the virus life cycle, and then to explore the virus dynamics inside cell and also to study the impacts of remdesivir on the virus. And then we want to integrate the results of intracellular model into a within host or intercellular model to study the viral dynamics in the body and also to study the impact of treatments with patients that they have different levels of immune responses. So from the point of the modeling, the intracellular model, the replication cycle that we are focusing on, important parts of, we are focusing on the key aspects of the virus life cycle inside the cell. One of them is the protein production or translation. The virus produces two types of proteins: structural proteins that the virus uses them to create new virons, and also proteins for replication process, which are known as non-structural proteins, These are the proteins that the virus uses to replicate itself, which means that the next step that we are including into our model is the copying of the genome or transcription. The virus copies itself through the transcription process. And during this transcription process, the virus produces some shorter RNA segments known as subgenomic RNAs, that these shorter subgenomic RNAs, they produces those structural proteins. And then we have the latest equation that we have in our model is the virion assembly from these structural proteins and the viral genome. And as you could see probably in this figure, the viral genome that has been shown in this dark green is a plus-strand RNA. It gets in a complex with the N protein or the nuclear capsid protein. It creates a helical structure forms, and then this helical structure gets covered by other structural proteins known as S-spike. Probably you know about spike. We are hearing a lot about it in the news. This spike protein is a target for treatment and also a target for vaccines. And then there are other proteins, envelope proteins, and then membrane protein M. So these reactions, all this has been included into this reaction. And then the level of all these S, M, E, and N in each particle, they have been measured experimentally. So we are using the experimental observation to make our model more realistic. So then now we have all the reactions. So I have included all the reactions into a model which contains more than 100 reactions. Then I use Gillespie algorithm to do a stochastic simulation of the model. There are some parameters that we know them from literature that has been measured previously, but there are some parameters that we don't know. What I did, I studied the impact of changing those parameters on the dynamics of the virus. In this slide, I will just focus on two of them: the speed of ribosome and the speed of transcription. Speed of ribosome is because the virus produces the proteins and ribosome does that. So what's the speed of the ribosome? What's the speed of producing proteins? We are seeing the impact of changing this speed from 2 to 5 to 10 to 15. And as you could see, increasing that speed will increase the number of total released virion from the cell. How I computed that? We did 200 simulation, and then this is the mean over 200 stochastic simulation of the model. And as you could see here in this small, after the infection, there's a time lag, there's a latent period that the cells get infected, but does not produce the virion and then starts releasing virions. And as you could see, increasing the speed of the ribosome, increasing the speed of production of proteins will decrease this time lag. So will decrease the time lag and also will increase the number of virions. This figure shows the speed of transcription. So the virus needs to copy itself inside the cell. What's the speed of copying itself? And we are seeing that increasing that speed will increase the number of virions and also will decrease the time lag. But the main point that we want to get from here is that although we don't know these parameters, and change these parameters will change the time lag and will change the number of virions, but the whole point is that the infected cells, they get infected cells after a time lag, they start producing virion almost linearly. This is something that we're going to use in our intercellular model. And this is, now I'm going to focus on two parameters that they are really important for us because they could show the predictive power of our model. These are ribosomal frame shifting probability Q and RTC elongation probability. So what's the ribosomal frame shifting probability? This line shows the genome. When the RNA comes inside the cell, it needs to produce proteins and then uses those proteins to replicate itself. So the ribosome that is inside the cell will bind to this open reading frame 1A. It's we say that there's just like a motif on it that the ribosome binds to that and will start producing protein. When the ribosome gets to another open reading frame, now at this point, the ribosome will make a decision. Do I want to stop the transcription, the translation? If it does, it will produce a protein known as polyprotein 1A, PP1A. Or if it can does a -1 ribosome frame shifting and keeps going, in that case, it will produce another protein 1AB. The virus needs both of these two non-structural proteins to create another protein. These two proteins will get cleaved and then binds together to form another protein known as RTC, replicase transcriptase complex. And virus uses these proteins to copy itself. So that's like a copy machine for the virus. And what this copy machine does, that also does something interesting. So the RTC starts the copying of the genome from the other side, from 3' ends. If it starts copying the genome, goes forward until it gets to some motifs known as TRS, which are gets to one of these motifs. So now here the virus will make a decision. The RTC will make a decision. Do I want to stop or do I want to keep going? If it stops the process, it will produce some small RNAs known as subgenomics RNA, which the virus needs them because, as you remember at the beginning, I said that the virus produces structural proteins from these short segment RNAs or subgenomic RNAs. And or if it says, okay, we just want to keep going, there are 9 TRS over the genome. So making this decision will happen 9 times. So we could have 9 subgenomics RNAs, and if each time it says, oh, I want to keep going, I want to keep going, it will replicate the whole genome. So we would have 9 subgenomics RNA, or we could have a replication of the whole genome. Transcription can give us like 10 RNAs, 9 subgenomic, and then 1. The whole depends which time the RTC wants to stop the transcription. By considering that, now we want to see what's the impact of changing these 2 parameters, the probability of ribosomal frame shifting Q and the RTC elongation probability R. What happens in our model if we change these 2 parameters? And as you could see in this figure, by increasing R, by increasing the RTC elongation probability, the time lag that the virus needs to produce its first virion is decreased significantly, which shows the virus, the RTC prefers to keep going forward most of the time rather than breaking the transcription process. But something interesting happens with the Q, the ribosomal frame shifting, and we see that if the ribosomal frame shifting is really low or if the ribosomal frame shifting is really high, that doesn't an optimized position for the virus, which makes sense because virus needs both proteins, PP1A and PP1AB, to make an RTC and then use the RTC to replicate itself. Our model predicts that the optimal range for the Q is something between 30% to 50%, which is in a really good agreement with biological observation on different coronaviruses that they have shown that it's something between 25% to 45%, and there are some other works that they have suggested this could be even a bit higher. So this shows the predictive power of our model, that we could predict a range that is in a really good agreement with biological observations. So now that the model acts nicely and predicts things in a really nice way, so now we want to study the impacts of remdesivir, which is a treatment has been developed recently, and not recently because remdesivir is a drug that they have been used previously for Ebola, but now they have shown that this also works for COVID-19. As here, the black curve shows when there's no treatment, so the cells, and we are showing the total number of released virion again here. The black curve shows when there's no treatment, so everything happens. And then the magenta shows when, here we are putting 25 molecules per cell, but then we're gonna increase the dosage to see what happens. The magenta shows when we start treatment after 50 hours, the green shows when we start treatment after 30 hours, and then 20 hours and 10 hours. And as you could see here, the earlier we start a treatment, the better results we get. Starting treatment during latent period will give us a better result rather than starting it later, like after 50 hours or after 30 hours. Also, whenever you start a treatment, it will flatten the curve, the number of total release virion will be reduced, but it would be more effective to do it earlier. In this figure, we are studying the impact of when we double the dosage of the remdesivir. What happened when we double the dosage? You see, for the 10 hours, we see a very, very better results compared to having 25. It will reduce the numbers even more. But regarding to 50 hours, we are seeing more or less same results. The dynamic doesn't change that much, which shows the timing for treatment is more important than the dosage, at least from the point of view of intracellular modeling. Now we have that result from our intracellular model, that when there's no treatment, there's a time lag, and then the virus releases viron linearly. And also we know what happens when we start a treatment. Treatment is more effective during latent phase than when the virus is productively infected and is producing virons. For our within host model or intercellular model, we have considered 6 populations. These are the uninfected cells or target cells. These are the lung tissue cells that they are living their life, they are doing replication, they are doing everything. And then when the virus comes, they get infected. So first they move to the latent phase, the phase that they are infected, but they are not producing virons. Then they move to productively infected phase. At this stage, they produces virons, and this viron will make more cell infected. So this cycle will happen. But due to the presence of viron, due to the presence of antigen, foreign antigen in the body, the antibodies will kick in, and then the antibody will start replicating themselves at a rate RA, and then they will neutralize the viruses that they are in the body. The other aspect of immune system is the effector cells, which could be like those T cells, or even some parts of some of the like natural killer cells, those other effector cells that they are the other part of immune system. There's already a small level of them, but that level is not enough for suppressing an infection. Due to the presence of infection, these infected cells, these effector cells will get expanded at a rate alpha. So alpha is the expansion rate or the increasing rate of effector cells. And then these effector cells will remove the infected cells from the tissue at rate mu. So we have 3 important parameters. We have RA, which is the replication rate, which is the proliferation or increasing rate of antibodies. We have alpha, which is the proliferation rate or increasing rate of effector cells, and we have mu, which is the removal rate of infected cells by effector cells. But again, there are some parameters that we know them from literature, so we use those parameters, but there are some parameters that we don't know from literature. What we did, we decided to fit our model with 12 patients' data. These are the first 12 cases that they were in Singapore. So what they did, they measured the level of virus in their body over the course of 30 days until they were feeling better. And the blue dot shows the level of virus in each patient at different times. What we did, we fitted these data to our model, and the red line shows our model prediction. As you could see, for most of the patients, the virus shows a 2-peak behavior. The level of virus goes up, comes down, goes up, and then comes down, which shows those when some people that are infected, they said, oh, I was feeling better, and then again I felt worse, and then everything went away. This shows this two-peak viral load behavior, which has been captured in a really nice way by our model. So now that our model captures the viral dynamics in the model in patient, we wanted to see what are the predictions that our model can do, and are these predictions in a good agreement with biological observations or not. So in this short table, I'm just showing that, for example, for latent period, our model predicts that the latent period for these 12 patients is something between 27 plus and minus 1 hour, which is in a good agreement with experimental observation that they have been done in a really different way through experiments. Regarding incubation period, so we are predicting that incubation period is something around 5 days, and is in a good agreement with experimental observation. And also for AAA is the antibody appearance day. Someone gets infected, after a while, the level of antibodies goes high in the body, and they start neutralizing the viruses. So the experiments suggesting that this is going to be something between 10 to 20 days, and what our model shows in a really good agreement with the experimental observation. So we just fitted the virus load to our model, but then all the prediction that our model is doing is in a really good agreement with the experimental observations. So now that the model is working, is doing the viral load, is making prediction that they are in a good agreement with the biological observation, we are considering different cases of immune response dynamics. The black curve shows a healthy immune system. These 12 patients, they didn't need treatment, so they were, we could say that they were a healthy case. They were someone with a healthy immune system that got rid of the infection over a period of time. So the black one is when we are using the median value of parameters from those 12 cases. And as you could see, for the level of viral load goes up, comes down, goes up, and then goes away. That normal to peak behavior. The tissue damage is not that much high. The level of effector cells goes high, but then comes down, and the antibodies comes in at the end, and then everything is gone. So now we're gonna consider the case that someone is immunocompromised. As you remember, we had 3 important parameters. We had mu for the removal rate of infected cells by effector cells. We had alpha, which was the proliferation rate or increasing rate of effector cells. And also we had RA, which was the proliferation rate or increasing rate of antibodies. So one case to be immunocompromised could be that when someone has a low mu, when the removal rate is reduced. The red one shows when the mu is low, the virus stays in the body for a longer time. The virus kind of tails off for a longer time. The damage would be a bit more, but not significantly. But what we see for effector cells is something really interesting that you normally don't see unless you're doing the models. And as you could see here, the level of effector cells or T cells, the natural killer cells, those cells goes to a really high number and then comes down slowly. As you could see, after 20 days, the level of virus is quite low. The damage has been is recovered, but we are seeing a high level of effector cells, which is a bad thing for respiratory diseases. Probably you have heard about the term cytokine storms a lot in the news recently. That's what happened when the level of effector cells, when these immune cell-mediated immune response still show a presence inside the lungs, that will cause having still the symptoms. Although the virus is gone, the virus is under detection limit, but people still can have the symptoms. So now we know which parameter can cause having those symptoms even after getting rid of the infection in the body and still having like a cytokine storm. The blue one shows another immunocompromised case when the effector cells are increasing in a slow rate. The proliferation rate is reduced, alpha is reduced. So which makes sense, when alpha is reduced, we are not seeing a lot of effector cells, but the level of virus goes to a higher value and then it goes down slower. And we see a much bigger damage inside the tissue. As you could, again, we could just look at the 20 days. What happens after 20 days? The virus load is low, the 2 peaks has happened. The effector cells is low, but we see a big damage. Still, there's a big damage that needs to recover over time. And this one shows the other case of patients that they have been recovered. They do the test and the result is negative. They don't have, but still they have problems with, like, they say we couldn't run like as we used to, we couldn't do the exercise. This shows that still the damage is in their body. So we know that which parameter is probably weakened in those cases. And the magenta one shows the case that the proliferation rate of antibodies is low. And as you could see, something really interesting happens here. We could see a third peak in the viral load in this case, which shows those patients that they are infected for a longer time. This shows that for those patients, the antibodies are not coming that much soon. The antibodies proliferation rate is slow, or the level of antibodies is really low in their body. And in that case, they could show a third peak or even the fourth peak in the level of virus in their body. And this will cause to have effector cells in the lung for a longer time, which could cause, again, something like a cytokine storm. And this shows the importance of antibodies. This is, as you could see, all these peaks were gone because of the antibodies at the end of the day. When they are not there, this will keep staying, which shows the importance of vaccine, because everyone is talking about vaccine now, because they know how important is the antibodies, because you see at the end things goes away because of the antibody. When antibody is not present, it's coming later. We are seeing the virus in the body for a longer time. This slide, now I'm gonna focus on doing treatment in those cases. So we have the black, which is the healthy. By healthy, we mean someone with a healthy immune system. We have red, when the mu, the removal rate is low. We have when the alpha, the proliferation rate is low, and then when the RA, that the proliferation rate of the antibodies is low. And you're gonna look at the impact of starting treatment in these patients at different times. Like dash shows when you start treatment after 7 days post-infection, dot line shows when you start treatment after 6 days, and dash dot when you start treatment after 5 days. And the green line shows the detection limit. So what happened, as you could see, all these figures, they are suggesting one thing: starting treatment earlier is more effective. Doing treatment in 5 days is more than 7 days. 7 and 6 in this, they are more or less similar. They come on each other curve. So there isn't that much difference between doing 6 or 7. But here we see the difference. But something that we see, starting treatment earlier is always more effective than doing it later. But there's something really interesting that we could observe here. And that one is that we know this is the detection limits. When the level of virus is under this, means test gonna show negative. But that's the importance of having a model. The model can show that the virus is staying in the body for a longer time. These people probably could infect other patients, other people, but the detection, they are under detection limits, and they seems fine, at least from the point of view of testing. Now I'm gonna focus on each specific part to see what from just now. Let's just focus on the level of virus load. What happened? There's something that it's obvious here, is that starting treatment earlier or later, you're gonna get rid of the second peak. The second peak doesn't matter if you do 6 or 7 days, the second peak is gone. Doesn't matter if you have a healthy immune system or you are immunocompromised. In all cases, the second peak is gone. But for the case that the RA is low, we see that the level of virus could stay around that detection limit for quite a while when you start treatment earlier. And these are when we are talking about risks. This is one of the risks that we are talking about, that doing a test will show that the person is fine and the person is receiving the treatment, but actually the person has the virus in his body and can make other people infected. So these are risks that we have to consider in our mind by doing treatment earlier. And now we're going to focus on the tissue damage, the level of T, the level of healthy cells. And as you could see, treatment always will reduce the tissue damage. Even in this case, when the RA is low, even doing treatment after 7 days, we are seeing that at least this part is gone and the body is recovering itself faster. But the earlier we start the treatment, the better results we are getting. The earlier treatment will reduce the damage significantly compared doing it even 1 or 2 days later will have a significant impact on the level of tissue damage. So in this figure now I'm gonna focus on, because we have that healthy immune system case, that black curve, that we say, okay, now if this person would have started treatment, what would have happened? So of course, doing treatment will reduce the level of effector cells. Doing treatment will reduce the tissue damage. Doing treatment will help to get rid of the second peak, but doing treatment does not always reduce the duration of infection. As you could see, doing treatment after 5 days, still the virus is inside the body more or less the same amount of time. These are the risks that we have to consider by doing a treatment earlier. That doing treatment earlier does not always mean that the duration of the infection is reduced, because we don't know who has a healthy immune system or who doesn't have at the beginning. We see for healthy immune systems, doing treatment does not always reduce the duration of infection, at least significantly in a way that we could, but will help the recovery. Less tissue damage, less level of effector cells, probably they will feel much more healthier, but they could infect more or less the same period of time other people. And now we want to consider one of the immunocompromised cases, the blue case, when the increasing rate of effector cells, the alpha was reduced. And as you could see, starting treatment regarding the tissue damage, starting treatment after 7 days, 6 days, or 5 days. So the earlier you start treatment, the less tissue damage you're gonna observe. regarding effector cells, the earlier start treatment that you start, the less level of effector cells you're gonna observe. But this doesn't mean always the earlier treatment means the less duration of infection. As you could observe here, when we start treatment after 5 days or after 6 days, 6 or 7 days, you see that after, like, say, around 16 days, the virus is under detection limits. But now still, when you start treatment after 5 days, still the level of virus is quite in a high number. And this is another risk that we have to consider by doing an early treatment. Early treatment will reduce all those damages, but the people that are receiving treatment could infect people, other people, made in a more effective way than receiving treatment earlier. But this doesn't mean that we have to start treatment earlier, because if you consider the area under this curve and area under the other curve, this parameter known as AUC, area under curve of viral load, it's a parameter that is being used to measure how treatment being successful or not. And we see that for the 5 days, this area is reduced significantly. So it shows that doing it earlier is more effective for that patient. So less AUC, less damage, less effector cells. So that person gonna feel absolutely fine, but now there's a risk that that person can infect other people for a longer period of time. By now, I'm gonna just in the last step, I'm gonna focus on doing treatment on those 12 patients that they were the first 12 patients in Singapore. So we have the data, they didn't receive any treatment. But now I'm gonna consider what would have happened if we would have started treatment on those 12 patients on the day that they had the onset of symptoms. When they said, we have a symptom, they come there, they do a testing, they say, okay, now you're infected. What would have happened if they would have started treatment by remdesivir? 8 out of 12 patients, as you could see, like patient 2, patient 3, patient 5, would have seen a significant increase in the number of virions and the duration of the infection. So they would have really enjoyed getting that treatment and having a short period of time. But some other patient, like patient 1, which is more or less similar to our median case, the duration of infection would be more or less the same for that person. Of course, less viral load, less AUC, less tissue damage, less level of effector cells, but the same duration of infection means that this person could cause a risk to the society for the same amount of time to make other people infected. Or patient 9, patient 11, patient 12 would show the same behavior. Considering someone like patient 7 probably would say, oh, I wouldn't gain anything from this treatment. So these are factors of this treatment that we have to consider, that it is important when someone is immunocompromised. In many cases could be effective, but does not always reduce the duration of infection. Another interesting thing that happens with treatment is what would have happened to the patient 4 if you would have started treatment after the onset of symptoms. And we could do that because for these patients, we know the exact day that they had the onset of the symptoms. And these are lucky patients. They were the first ones. They did all the measurements on them. So they were lucky patients, you could say. In this case, we see that the level of virus, even after treatment, would have gone up and then come down, maybe even a bit slower compared to no treatment. But you see the peak is reduced significantly. The area under it is reduced significantly. But as I said, the risk that we are considering now for all patients is that the duration of infection would be more or less the same for some cases. And we don't know, for example, the day that they have symptoms, like patient 2 and 4, no one knows what would have happened by starting treatment. And if we started treatment and things are going up, this doesn't mean that, oh, the treatment is not effective on this patient. It is effective, but it takes more time compared to other patients. And in conclusion, what we have observed from these 2 intra and intercellular models, our intracellular model showed us some new insights to the intracellular process of the virus, such as those ribosomal frame shifting rate Q, which we could predict something which is in a really good, nice agreement with the biological observation. Our intercellular model, by fitting our intercellular model to patient data, we could observe that second peak in the level of viruses, and then we could see that the level of tissue damage, the level of effector cells, it really depends on the level of immune responses. And an integration of these two models helps us to study the impact of treatment. And we observe that the earlier we start the treatment, the better result we get. Earlier treatment means less tissue damage, means less viral load, means less level of effector cell, which is a bad thing for respiratory diseases. But this does not always mean less duration of infection. And something that is important, immunocompromised patient, you probably remember that blue one. The blue one really enjoys the case to get a treatment, but the black curve would have been more or less the same duration of infection. So we could get to this point that the immunocompromised patient, they get more benefits from treatment by remdesivir than a person with a healthy immune system. So people that they are healthy, by healthy, I mean that they are athletic, they have a good immune system. If they want to get a treatment, they have to maybe think twice, because this drug has significant side effects. So people should be really careful about getting that treatment. And at the end, I want to thank the Wellcome Trust for funding this project, for funding my postdoc, and also Ryden, Richard, and Eric for their supervision. And thank you for your attention.
Thank you very much. Thank you for that. Fantastic. So thank you to all speakers. We probably have